Hello, and welcome back again to the Royal Institution. Again, it's very nice to see faces that have become familiar in the course of this series. Now, so far, at the molecular level, which is where all the chemistry is, we've only been in the land of Lilliput. And it's now time for Gulliver, for us all, to move on to the land of the giants, Brobdingnag, at the molecular level. We've seen pygmies in action, like nicotine and adrenaline, and they only have about 100 atoms. They're pygmies. They're the miniature molecules. Now we want to talk about molecules that have got millions of atoms in them. And our lives, as you will see, are literally in their hands. Our lives depend upon giant handed molecules, because of course the theme of these lectures has been handedness. And so today we're going to concentrate on three. First of all, the carbohydrates, and then we're going to look at proteins, and then we're going to look at the ultimate handed giant, DNA, deoxyribose nucleic acid. Now, the simplest of these three are, in fact, the carbohydrates. They tend to get a bad press because you're always reading, you have too much carbohydrate, you get fat, and you must knock off the pasta, and so on. All right, but in fact, remember that the carbohydrates are the sources of glucose, and glucose is the energy carrier of our body. And what I want to concentrate on just to start off today is the carbohydrate which is called cellulose. And it has a very important function which we tend to forget about. What I've got here is an instant miniature Royal Institution jungle. Now, what it consists of, you'll be amazed to see, are plants. These are plants, okay? And remember that plants cover the surface of the earth. Well, I suppose except for the Sahara Desert. But anyway, there are lots and lots of plants around the place, and they're being held up. That's not just an accident, that's scaffolding. That is carbohydrate scaffolding. But you can't always see, of course, the cellulose skeleton. By looking at a tree, you wouldn't say, oh, well, it's got a cellulose skeleton. So let us sort of bring that to life a little bit. We have at enormous expense, because they're out of season, here a vegetable marrow. And that vegetable marrow is mainly water, because inside that marrow, and responsible for its shape, is a cellulose skeleton. Cellulose. A loofah. You're familiar with loofahs? Do you scrub your back at night? Well, if you do, you could use a loofah, I see, like that. Very, very good, very tough, durable, strong skeleton. Now, I've said about strength. What we are doing is actually using cellulose for its structural properties. Let me describe for you another cellulose version, which again we use for its structural properties. Cotton wool. Right, if I take out a nice chunk of that, We're all used to cotton wool, jolly good for cradling things, and it's all made of fibers. That's all extremely familiar to us, cellulose fibers. And if we twist this, I'm a rather inefficient at this. If you do that efficiently by machine, then that is the thread that you get. You get a cotton thread. And that's a useful thing for sewing up things and holding things and so on. Very important structure. And then we can think, okay, but, well, that's not much good, is it? It's not very strong. That's a rotten piece of thread. But what I want to try and impress on you is the fact that it's actually a bit stronger than it looks. That's a rotten little demonstration. So let's have a competition. We're going to have a molecular competition. Very simple. Thank you very much. Here's the arena for this competition. Now, can I have a couple of volunteers? Thank you very much. I need a team, you see. Thank you. Please come down. What's your name?
Simon.
Simon. Hello, Simon. And your name?
Victoria.
Victoria. Okay, jolly good. Now, I want one of you to be cotton, and so you can be there. Okay. And you can come this side, Victoria. Okay. Now, let me just explain what this extremely sophisticated demonstration is. Okay. What we've got here is a steel wire. Steel. It's made of steel. And on this side, we've got a wire which is made of cotton. Right, now, which do you think is stronger, cotton or steel? How do we test this? This is a Royal Institution tensimeter. That's a fancy name for it. Right, okay. Now, what we're going to do is successively load these wires with weights, which are hiding behind here. So will you put on a weight, please? Thank you very much. That's a 0.5 kilogram weight. Okay, so that's 500 gram weight on each one. Very gentle, so we don't perturb anything. And notice that the tensimeter is reading 500 grams. Right, okay, and both of them are holding. Let's have another one. Very good. Very good. We're now up to 1 kilogram. Tensimeter is reading that on each side. Exciting, isn't it? Right, okay, let's have another one. And, oh, it's gone. But his is holding. That's the steel one. So the steel one has gone first. All right? I tell you what we're going to do, Victoria. We're going to borrow one of your weights and give it to Simon. Would you like to try that on yours and just see if it'll take one extra? And it's still holding. Right. Thank you very much indeed for coming to help us with that. That's fine. So there must be something special happening in this cotton thread to make it so strong. And if we're going to find the reason for that, we must go, thank you very much, we're finished with that, we must go to the molecular level. If we want to understand phenomena, we must go to the molecular level. Chemistry. Right. Now I remember that I said that this thread was made up of glucose units all stuck together. Here is the glucose molecule. Now, let me just tell you about the structure of this molecule. First of all, this red ball is an oxygen atom. And the pale blue ball is a hydrogen atom. And if you look at this molecule, you'll see that that grouping, this hydrogen-oxygen grouping, is represented several times in this molecule. And it is actually the association of these hydroxyl groups, HO groups, together in the thread, which make that thread strong. These are sticky groups, and we're going to come back to molecular stickiness. Now let me just take you from the individual unit to a model of cellulose. here we are, a string of glucose molecules, and the sticky groups that we've just seen represented by these white dots. Okay? So now let's go up from this model, this molecular model of cellulose, to a piece of thread. Okay? And if we stretch that piece of thread down the length of that model, then what we are doing is representing the thread at the molecular level. And it is the stickiness between these chains of the hydrogen-oxygen grouping that we've just seen, which gives cellulose its strength, in this case, its tensile strength. It's a molecular association phenomenon. Right, now let's move on to proteins. What about proteins? This is the second of the giants. And proteins have got very many more jobs to do. You see in the carbohydrates, we've got starch, which is source. That's the pasta and the potato and all the rest of it. That's a source, a direct source for us of glucose. Cellulose, the scaffolding, and food, incidentally, for horses and cows and so on. We can't digest it. It's the fiber in our diet. But apart from that, there are rather few functional carbohydrates. Proteins are, in fact, much more versatile. They've got many, many more jobs to do in life. And how can we illustrate that? Well, let's take, rather than just have a dollop of soya meal and so on here, let's take a more living example of proteins in action. And it's very nice to have from the London Zoo. Hello, thanks very much for coming.
You're welcome.
Your name, please? Andy. And you brought with you?
Tyto.
Tyto. Isn't she beautiful? Isn't he beautiful? Isn't he beautiful? Tell us about Tyto.
Well, he's 3.5 years old. He's a European barn owl.
Yes, so common in Britain?
Unfortunately not. Only about 10,000 of these left in the wild.
Yes.
Very
beautiful.
Rare.
And how many have you got at the zoo?
Well, we've got breeding barn owls at the zoo, but this is a special one. He's hand-reared,
yes.
and we use him for talks to children, educational purposes.
Super. And very, very tame.
Yeah.
Feels beautiful. Now look at Tito. How is he showing off proteins? He's also extremely interested in the show. That's good. Look at his feathers. They're made of protein. Look at his claws. They're made of protein. And his eyes, which he's extremely alertly looking round about for. And remember that an owl's eyes are extremely good optical detectors, because he depends on dinner for what he can see in the dark. All right? The lenses of his eyes are made of proteins. And then, supposing he's had lunch, he might have a mouse inside him, okay, which is another bit of protein, incidentally. But the way that he'll be feeding on that mouse will be by using enzymes, and those enzymes are proteins. And so Tito is a lovely illustration of what can be done. Now, another very important, because you'll already be familiar with this, is muscle. And I nearly forgot to mention that. Can you make him flap his wings a little bit? Oh, lovely. You see, and what he's using is muscle to flap his wings with, and they're made of protein. Thank you very much for bringing him along.
You're welcome.
Lovely. Thanks. Now, once, of course, you've had an owl, you can guess what we're going to have next, can't you? What are we going to have next? Pussycat. You're quite right. Jolly good. Will you bring? Thank you very much for bringing. What's your name?
Karen.
Karen. Thank you very much for coming. And what is Pussy's name?
Millie.
Millie. Okay, Millie. Lovely. So we look at Millie. Now, let us think in scientific, chemical terms. What is Millie? Well, what she is she's a cuddlesome bundle of molecules, all right? And don't you forget it, she's all molecules, all right? And so her fur is molecular and it's a protein. Her claws, just like Tito's, are protein. And she may have something in common with the owl in having a mouse inside her. Did she have a mouse for lunch? No, nothing so disgusting. Okay, but whatever it was, she probably had a proprietary cat food, which I'm not allowed to mention, and she's digesting that also with her enzymes, which are proteins. Thank you very much for bringing her along. Thank you. Now, the proteins of these two animals, of course, are really more subtle than we can show there because, of course, all of their control mechanisms, their brains, the important parts of those, are protein. And that applies to us too, of course, and matters like our memories are matters of proteins. Now, where does this versatility of the proteins come from? They're much more versatile than the carbohydrates. Where does it come from? And again, we must look at the molecular structure in order to answer that question. So let's have a look at a protein molecule, which I just happen to have here. And I found the end. Yes, that was good. Let us just uncoil it. And I want you to have in mind the comparison with the carbohydrate molecule that we looked at before. I'll just take a coil like that, and that's fine. I'm coiling it up a little bit because that's the way in which many proteins do in fact exist. Notice it's got an end there. And when we look at this protein molecule, what do we see? First of all, we see that instead of all the units being exactly the same as they were in the carbohydrates, that here they're different, they're all sorts. We've represented this by having different colors and different shapes. And what we have here, in fact, is 20 different possibilities in our chain of units. In the carbohydrates, like cellulose, our chain unit was glucose. In the proteins, our chain unit is an amino acid. And as I said, there are 20 of those, and they can be arranged in these chains in any order. In the case of a molecule like cellulose, we have about 1,000 glucose units, but in proteins, we can have up to 100,000 of these amino acid units all linked together to make our protein. And one other point about these proteins made of the individual amino acids. All of the amino acids in the proteins of the owl's feathers and the cat's fur and our skins and muscles, they're all left-handed. And that's actually quite a striking point, and we're not at all sure why that is. We can make all right-handed proteins in the lab, but that's different. The natural ones are all left-handed. They're big molecules, as we've seen, and they've all got specific jobs to do. They're not all jack of all trades. They're highly specialized target molecules. So let's look at the first enzyme. The first enzyme I want to mention is lysozyme, which has a very special connection with the laboratories here in the Royal Institution. And here is a model of this enzyme, and in fact it was the first enzyme to have its structure measured. And that was done by Sir David Phillips and his students here in these laboratories. Now, what is lysozyme anyway, you might ask? Well, I can tell you that literally in the twinkling of an eye. When you blink, your eyelid is like a windscreen wiper. It's wiping your eyeball. But then think what your eyeballs are like. They're warm and wet, like British beer. And that's an ideal place for bacteria. It's really a sort of bacterial rest home. And they can settle down in your tears and they'll breed like crazy at your body temperature. And so the body has got to protect itself from these foreign invaders, and it does this with lysozyme. Lysozyme attacks these bacteria. Now, that's the model, but let's look now, and that structure was determined in 1964. Let's look at a modern computer representation of that structure. And I've got a highly trained computer operator. Yes, thank you very much for coming to join us. What's your name?
Tim.
Tim. Okay, Tim. Now, Tim is on instruction going to produce pictures for us, but he's going to let us just have a quick look at this picture. So here is lysozyme, and you can see the resemblance between this structure and that structure. Okay, the structure we've just been looking at. Furthermore, this structure shows us the gaping jaws of this molecule, ready to bite. Exciting, isn't it? Okay, now, what we want to see is this structure turned into what is called a space-filling representation, where we're going to put balls representing the atoms all over these sticks. Can you do that for us now, Tim? Thank you very much. Now, it takes a little bit of time because it's actually quite a complicated computer change. And what I'd like to just emphasize is that this method of looking at molecules has become very important in chemistry, because we don't necessarily need to go dashing off into the laboratory to look at what reactions we might do or how molecules would fit together. We can explore that on the computer because we can move molecules into their neighborhoods, see how they fit and what they might do, and then we can go off to the laboratory and do our experiments more directly and more economically. And there we've got it. Thank you very much. So we can see the mouth now of this enzyme very clearly here. And notice the huge number of atoms that it's got. And it needs those in order that its mouth has the right shape at the right place and at the right time. Tim, thanks very much indeed for coming to show us that. So what happens? What happens when lysozyme spots a bacterium? Well, we can represent that. Here we have lysozyme's mouth. Okay, this is just a model, of course, of the shape of the surface. And here we have a bacterium planning to invade your body through your eyeball. And if I now allow lysozyme to grasp the bacterium, you can see what it's done. It's broken the cell wall, and that is actually what happens. This red part is the cell wall of the bacterium, and it breaks and falls. So that bacterium is now a dead duck. That's an appropriate way of putting it. It's not going to do you any more damage. It hasn't got a cell wall, and it can't operate. Right, now let us turn to another enzyme. And this is the enzyme catalase. Catalase. Now it, again, just like lysozyme, has got a specific job to do. And the specific job that it's got to do is to turn hydrogen peroxide into oxygen and water. Now you might say, why do we need to bother to do that? Well, hydrogen peroxide is produced all over our bodies, and it's important that we get rid of it. It's a byproduct of our chemical reactions, and it's important to get rid of it. And so catalase is sent to get rid of it. And we've got catalase in most parts of our body. But it just happens there's a lot of catalase in liver and So we can use liver to show the source of catalase. Now what we've got here is just hydrogen peroxide, aqueous hydrogen peroxide showed, okay? And I'm going to put some catalase in it, okay? Teaspoonful chopped liver, rich in catalase, and in we go. All right? Notice that already we get bubbles off. Bubbles. Can we show what those bubbles are? Well, this is a lovely, nice piece of chemical nostalgia. And you've probably done this yourself at school. You light a splint, get it good and alight, and then blow it out so it's still glowing. If we put it in there, it rekindles a glowing splint. Right, it's the classical test for oxygen. It gives any chemist a great deal of pleasure. So much pleasure, I'll just do it a couple of more times for you. Can't resist this one. Look at that, isn't that marvellous? Just do it once more, just for fun. Oops, there we go. So that's catalase on the job. Very fast and very snappy. All right, now let's go into the laboratory. Because one of the things that chemists are always trying to do is to do better than nature. It's a challenge, okay? Can we now heat our laboratory specimen? Thank you very much, Bryson. This is our laboratory specimen, exactly the same hydrogen peroxide. And just to sort of try and be convincing about this, we're really heating it, right, to see if it'll decompose, right? So if we light our glowing splint now, make our glowing splint, there we are, well alight, plenty of glow, dip it in, what's going to happen? Nothing, all right? Just to be absolutely sure that everything's all right, relight, glowing, in we go, nothing, all right? So there's obviously between these two processes, the one catalyzed by the enzyme and the other being just done in the laboratory without any catalyst, there's a huge difference. Now, I can't wait long enough. I can't wait long enough to do any more for that. So, what I want to do now is, we're getting that, right? Yes. See, that's. Make out what was going on. Right. Now, we've obviously got a big difference in rate there, because the rates of reactions are very important, particularly with enzymes. You can't wait about while you die of hydrogen peroxide poisoning. Catalase has got to get in there and zap it. All right. Now, how can we deal with time? How can we measure this time scale? Well, in the Royal Institution, they have extremely sophisticated methods of measuring time. And here we have, here it is. This is the Royal Institution hourglass. And of course, what happens with hourglasses is that sand runs through. Now let me just imagine this for you. If every time a grain of sand goes through into the lower bulb, catalase has sprung on hydrogen peroxide and zapped it, right? The comparative rate of that reaction done in the laboratory is not to allow one grain of sand to get through, which is the catalase rate, or even this bulb full of sand, or even a sack of sand to go through. The amount of sand that would have to go through would be 150,000 metric tons. And that is more sand than this entire lecture theatre full would hold, even with you lot in it. Now that's just been really a brief tour of these two giants. And now I want to go on to the third giant, the marvellous DNA, in whose hands we literally live, because it controls our lives. And so it's high time we looked at it. Indeed, DNA is the instruction book. It's the workshop manual, the blueprint of life. It essentially controls everything that we do and everything that we feel. It makes our proteins and it determines our heredity. It determines what we hand on to the next generation. Now, where is it? Where is all this DNA that he's going on about? Well, I'll tell you. It's in the nuclei of our cells. Of course, nucleic acid, that's where it gets its name from. Here are the chromosomes in our cells, and we've got 23 pairs of these chromosomes. So we've got 46 altogether. Let's just have a close-up of a chromosome, and here we are. This is coiled up DNA in our chromosomes. Right. So what's the nature of this marvelous molecule? What is it like? Well, it's been known for a long time that it's in the nuclei of cells, but it's really only quite recently that we've known what this powerful molecule is. And its structure was determined by three people, Crick, Watson, and Wilkins, in the 50s. And this is the greatest biological discovery of the 20th century. Here is a little model which I can make some points with, and this model was constructed by Crick and Watson when they came to this very lecture theatre to talk about this stunning discovery. How much have we got of it, first of all? You might ask. How much of this magnificent molecule have we got? Well, I'll tell you, in each cell of our bodies, we've got a 1 meter length of DNA. 1 meter. Okay, that's a very, very long molecule. But the other thing is that we've got 60 million cells in our body. And so if you take all the DNA out of our cells and string it all together, the length of that piece of DNA would stretch from the Royal Institution to the Sun and back again 1 million times. So it is a stupendous amount of biological material. Now, the other obvious thing that I want to tell you about it, it's a little spiral. It's a double right-handed spiral or helix. It's a little magical staircase. Now, it's difficult for me to explain the details of this molecule without looking at the father of this model. And I think Crick and Watson would have been proud to have this model. Let me just say a little bit about its structure. First of all, as we've seen, it's a double right-handed helix. It's a staircase. And while we saw that, for example, the carbohydrates were made of a single unit, repeating glucose all the time, like cellulose, we saw that the proteins were made of a single type of unit, the amino acid. DNA is again a polymer, just like the other two, but it has a 3-unit makeup. And I want to explain the 3 units. First of all, it has a sugar, and the sugar is deoxyribose. It's a cousin of glucose. It's very similar to glucose, and there it is. Each of these on this double helix is a deoxyribose unit. The banisters of this magical stairway are made of sugar. And the sugar units are linked together by phosphate, okay? These bends here represent a phosphate unit. There are 2 in each link, okay? That phosphate is just the same as the calcium phosphate of our bones or the phosphate you put on the garden as a fertilizer. So we have a spiral staircase which is sticky. It's sticky because we've got those hydrogen-oxygen groups all over them. And we'll come back to that point in a moment. Right. Now, the other point about this spiral staircase are the steps. The yellow part are the banisters, but these coloured parts are the steps. And these are called bases. We don't need to worry about the nature or the structure of the bases for our purposes. But the bases have two very important properties. First of all, they themselves are sticky in the same sort of way that sugar molecules are sticky. And what they do is stick to each other. Let's take this step or this step. Let's take this step, for example. Okay? Then we've got these lines here represent this sticking together of the bases on two separate banisters. And the effect of this stickiness is to pull the banisters together and give this molecule its shape. That stickiness imposes on this double, on the structure of this molecule, its double helical structure. That's one point about the bases. And the other point about the bases is that these steps of which the DNA molecule is made carry a code. There are only 4 different types of bases, and we've illustrated in this model. They have the initials T, A, C, and G. So there are only 4 of these. And what happens is that all of these steps are made out of either T and A or C and G. And the order of these steps in this spiral staircase determine the order in which the amino acids are linked together to give our proteins. And so that's why DNA is the code book for making proteins. And so you can imagine that in a certain length of this DNA, and let's think of a length which might be, say, 50,000 steps. That's a jolly long staircase. 50,000 steps, but a whole DNA molecule has got millions of steps. Let us just think of 50,000 steps. Those 50,000 steps could say in the process of protein manufacture, you will have blue eyes, you will have black fur, you will make catalase. And these bits of the DNA, if you like, they're like pages of the instruction book. These are called genes. Okay? And so we have the gene for making this, and we have the gene for doing that. And that's a very important matter. Now, the whole collection of these genes, which are spread all the way along the DNA molecule, the expression for that, this collection, is the genome. Okay? And that's an expression that's bandied about a lot these days. So the genome. So then we can ask the question, well, how big is a genome? Well, let's look at that question. Let's look at the size of a genome. And you're going to show us, aren't you? What's your name?
Rona.
Sorry?
Rona.
Rona. Okay, Rona. Come here, right? And what we've got here is the genome of a single cell organism. This organism is called Escherichia coli. And you all know Escherichia coli very well. You may not know intimately, but you've all got Escherichia coli inside you at the moment. It's living in your guts. But let's show them then this little bug, only one cell, its genome. Shall we hold that up at the edge there? You hold at that edge. Can you read upside down? Because if not, just come around that side for a wee moment. Okay, that's fine. Let's just read off a bit of this genome. Wherever you like. Just read. Start. Yes. Yes?
TGA, TTE,
Yes.
TCG, GTG, AAA, GGC.
Isn't it boring? Thank you. Now, you can see how boring it is. If she went on reading the whole genome, she'd be here to the next century. And that's only for a one-cell bug. Let's just give them an idea of how big that is. Go on. Pull, pull, pull. I'll give it. All right, look at that. That's what it is. And that's just for one cell, a one-cell organism. So you may be thinking, in your superior way, well, if that's all it is for a one-cell organism, what about us? Rona, thank you very much indeed. We'll just fold that up like that. You may be thinking, well, what about us? What about us? What's our genome like? Well, it's not all that easy to illustrate for you, actually, but we're doing our best. And here comes the human genome in book form. Right? Thank you very much indeed. That's splendid. This is the sort of size that the human genome would be. 90 volumes, 3 editions of the Encyclopedia Britannica, all printed out in a 4-letter code. That's all the code. It's amazing code. We only need 4 letters to describe you, whether you've got blue eyes, brown hair, or you're 6 feet tall. That all comes from 4 letters. Right? So that's the size of it. It's a huge collection of information, but that's the instruction book. Now, before you get any sort of superior-like feelings about that, let me just bring you down to earth. If you were a salamander, you would need not 90 volumes of the Encyclopedia Britannica, you would need 30,000 volumes of the Encyclopedia Britannica. And then you say, well, why on earth that? Well, the answer to that is that we don't use an awful lot of our DNA. We only actually, every day, are using about 1%. And so a few other bugs can get the better of us, or simple organisms. There's no direct connection between the two. Now, if you've got such a huge code book, and that's what it is, that's all it is, that's just a code book. What about proofreading? If you make a mistake in the staircase order of DNA, then you're going to make the wrong protein. And if you start making the wrong proteins, then this may be a cause of cancer. And this can come about by damaging your DNA, by radiation, or by chemicals. The process is called mutation. That is, mutation simply means change. You're changing an order or changing a base on the staircase. And this can cause cancer. And that, of course, is a tremendous challenge for chemists, because one attacks that problem, the problem of cancer, which is so common, by chemotherapy. Chemists would like to be able to say, we want to attack that human problem. And so that is done. Cancer chemotherapy. Chemotherapy means chemical cure. Is very common now, but it's very difficult to do without causing after effects. And I came across a nice story just last week of some American school kids. One of their number had contracted leukemia. He was being treated with chemotherapeutic agents, but he lost all his hair, which is a very common symptom of chemotherapy, a common result, after effect, side effect of chemotherapy, very common. And so all the other kids in his class shaved their heads too, so that he wouldn't feel conspicuous. I thought that was rather nice. Okay, so there's always then, that little story tells us that the scope for development of chemotherapeutic agents. And I had a very recent example of this and a nice experience. I was at a conference in France, And there on the program was a very distinguished and very clever American chemist by the name of K.C. Nicolau, and he was talking about a new cancer chemotherapeutic agent that he had designed and built. And here it is. Well, what we want to do is to try and reenact the chemistry of this new chemotherapeutic agent for you. Now, we worked on, in trying to design this demonstration for you, Roger the Rocket, he came to be known in the process of development. And here he is. The way that this cancer agent, the cancer chemotherapeutic agent, as a curative agent, works is to attach itself to the DNA molecule of this rogue. This is rogue DNA. This is DNA that's gone wrong. Bryson, could I ask you to attach that? Because I'm bound to do it wrong if I don't let you. So this chemotherapeutic agent sits all comfortably in what is called the minor groove, that narrow distance between the staircase, between the banisters, that's the minor groove. The big one is the major groove. Okay, so it sits comfy as you like in, we're all balanced, very good, thank you very much. We're balanced in the minor groove of DNA. Now, what does this cancer chemotherapeutic model consist of? Molecule. What does the molecule consist of? Well, first of all, it's like a missile. You see, what Bryson has really done is to launch the missile at this molecule. First of all, it's got to stick. And the tail of the missile is made of sugar. It has glucose-like molecules stuck in its tail. It's a multi-component missile. So those are sticky, and as we've seen, the banisters of DNA are sticky, and so it's sticking on the banisters. It's like sliding down the banisters, but not sliding. Okay? So it's in there. Right. So that's the recognition part, which is so important. And then we have a warhead. It's that warhead which is going to destroy the DNA. That's what it's attacking the DNA with that warhead. Now, of course, we can't have missiles running around people's bodies with warheads that are going to explode just, at any moment. Can't have that. And so we have to have a trigger on the warhead ready to do the job. And there is the trigger. Now, at the molecular level, and this is just one of these chance coincidences, the mechanism of the trigger for Roger the Rocket, used by Professor Nicolau in his very nice work, was actually based on work that my students and myself did 30 years ago. We never knew it would be applied in such a delightful way. But we must look at the warhead. And what I want you to see is that this I'm going to tell you is a molecule. You're thinking, well, it's just a bit of Meccano. Wrong. That's a molecule. Okay? And if I do this, okay, I want you to notice two things. We're going to rearrange this molecular structure. And this is what happens when the trigger that we talked about, when the trigger is activated, what happens is this. Watch what's happening. As we pull those together, can you see we rearrange the structure of that mechanical molecule? And at this level, the mechanics of what I've done are exactly the same as what happens at the molecular level. Any chemists in the audience will recognize that part, but what we have done is that we've produced two razor-sharp molecular knives at each end here. Now let's see what that does to DNA. And what I'm going to do, are you on standby? What I'm going to do is I'm going to activate the warhead. All right? I'm going to activate the warhead by pressing the trigger. And when I stand back, look what's happened. That DNA molecule is not going to make any more wrong proteins. It can't. The code book, the pages, there are pages of that code book missing. It can't do its job. And that's what this chemotherapeutic agent does. Now, Professor Nicolaou sent us a very nice video of that happening so that you get a feel of what it's like at the molecular level. Here is Roger the rocket in atomic form, atomic and molecular form. Here we are. Here is the warhead. Here is the trigger. And there is the sugary tail, right? So the patient takes the pill. It's now circulating through the patient's bloodstream, seeking out its target. Exciting, isn't it? Okay, right, there she goes, off to seek the target. Here it comes back again, seeking out the target. Here is the rogue DNA. Watch the recognition. It's slipped into place, all comfy, and then bang, goes the warhead. We just let those pieces separate, and there we are. The DNA molecule has been cleaved. Now, that sort of chemistry is right at the cutting edge of what chemists are trying to do in their part in helping with the ills of mankind. Now I want to talk about meteorites. And you should be saying, what on earth is he going to say about meteorites that got any relevance to what we've been talking about? That's what you should be thinking. Right, meteorites. Well, you may not have realized it, but this Earth is bombarded every year by about 100,000 tons of stuff from outer space. 100,000 tons. Most of it arrives as sand, as a matter of fact, but a few bits arrive as really quite large lumps, like this. So when you're out walking, be ready to duck. Okay, so that's a big piece of meteorite. But that piece of meteorite is actually, from a mineral point of view, and I have to be careful what I say in this lecture theatre, is actually rather dull. It's sort of things like silica and iron and things. And it certainly gives us no information at all about organic molecules. But in 1969, in Australia, fell a meteorite which does give us some information. And this meteorite, here it is, that is the Murchison meteorite. And that meteorite was found to contain amino acids. Amino acids. The building blocks of the proteins which we've seen that DNA makes. And we've been talking all through this series about handedness. And we've been talking about handed molecules. And we've said that all the proteins that we make are made out of left-handed amino acids. And we can ask the question, well, where does this handedness come from anyway? Where did it start off? And we might think, well, perhaps it came from outer space. We've got handed molecules in the Murchison meteorite. Is that the reason? Is that the source? Well, I'm sorry to disappoint you. No, it isn't. Because the amino acids in the Murchison meteorite were half left and half right. It was a 50-50 mixture. And so we need to look elsewhere for the solution to that problem. It's still not solved. Many people are thinking about it and speculating about it. And in that connection, I want to finish on a slightly different note. There are lots and lots of things that we don't understand. That's science job to find out about. And I was reading the other day a poem by John McCrae. John McCrae was a young Scotsman who fought in the trenches in France in the First World War. And he wrote a poem called Flanders Fields. And this was published, and again it's another little irony, it was published in Punch in 1915 on December the 8th, which happens to be my birthday. And he was seeing in the trenches his friends being killed all around him. And he wrote this poem, and I just want to quote a couple of lines. "Take up our quarrel with the foe," he said. Take up our quarrel with the foe. His foes were the people that he had to fight against. But those are not our foes today. I would like to suggest to you that our foes are those of poverty, ignorance, and disease. And scientists can help a lot with all of those, as we've just seen. And then the other line that he put next to that one was, "To you, from failing hands, we throw the torch." Now, people like me at my age, I can't contribute very much more to science because time's against me. But you can. And so I say to you from failing hands, we throw the torch of science. Be yours to lift it high. And so if you go out from here and you like being curious and wondering about how things happen and the way things are and why they should be, then do science, because it's full of excitement and interest and enthusiasm. There's nothing like it. Go out and do that. And goodbye from the Royal Institution.